Okay, so we're just going to do a quick review of these X-linked or sex-linked Punnett squares. We start off here with crossing. You can see a homozygous dominant for tan individual with a male that shows the recessive trait yellow-bodied. So here's my male with the recessive trait, and here's my homozygous dominant female. And let's just jump down to what's the ratio of tan flies to yellow flies. Well, there's a capital T in every one of these. There's no way to get away from it with this homozygous dominant. It's everywhere. It's going to be in every individual. So every single one is tan. How many males in the F1 have the recessive yellow allele, which would be the little t? Here is what we're looking at. Find a little t there. There's none. There's none. So remember, too, that whether this is, in this case, the little t would give us yellowness, but in the Morgan's cross of eye color, the small w would give white eyes. We don't see them in this first cross. There are no white-eyed individuals in the first generation. And if a male had that one white-eyed gene, we would see them. So we would not only have it in their genotype. If a male had that recessive gene in their genotype, we'd see the trait. They would actually be yellow in this case, or a white-eyed male, and that did not occur. So none. Nobody has that. No males, anyway. So you may not have seen that, but whatever. No males have that. Are the females homozygous? They're heterozygous, and that is the big outcome here, that we know if we cross the white-eyed male in Morgan's original cross or the yellow-bodied male in this cross, we know, therefore, that this recessive gene is going into every female. Every female is going to be heterozygous for the trait, which brings us to our second trait. Now we're crossing that heterozygous individual. We can see there's a female with the recessive trait that didn't exist in the P generation, but now it does. What the big outcome here is, yeah, we get tan, tan, and tan. Three of them are tan. Anytime you see a capital T, you're getting tan. But in this case, small t with nothing to offset it in the Y, we get the one yellow individual. So what's the ratio? 3 to 1. We saw 1, 2, 3 tans and only one yellow. But the reason it's a 3 to 1 ratio is literally out of the males, it's 1 to 1. There's just as many yellow males as tan. In Morgan's original cross with the eyes, there would have been just as many white-eyed recessive males as red-eyed dominant. So then there's this other cross that Morgan does with the heterozygotes versus an individual showing the recessive trait. So now we're test crossing the individual showing the recessive trait. Remember, in the Mendel's crosses, this would have been homozygous recessive, but in the sex-linked crosses, it's hemizygous for the recessive trait. Fine, we do this, and if we cross the heterozygous female with this individual who's hemizygous for the recessive, we get this outcome where we're homozygous recessive for a female. So now we see the recessive trait in a female, and we're also going to have an outcome where we have males showing the recessive trait. So we end up with a 1 to 1 ratio, and it does show that if you do this cross, the hemizygote versus a heterozygote, you will get some females expressing a recessive X-linked trait. And clearly then, if this had been if we then took this individual, this homozygous recessive for the X-linked trait versus the male we're going to get 100% 100% yellow individuals, even though we didn't do that that cross today. So I hope this helps you understand and how that should have worked. Thanks.